So I want to welcome everybody here. I know today is a busy day with all the interviews for the graduate students, so thank you for coming. It's my pleasure to introduce Professor Alessandro Usiello. And he was born in Italy, I believe, but he did not do his PhD in Italy. He actually went to France to do that, and I'm not sure why, but he went to the Institute of Genetics and Molecular and Cellular Biology. I'm going to say it in the English way. And he has his PhD in molecular neuroscience. But then he went on to do 2 postdocs, and his last one was at the Karolinska Institute in Sweden. And he actually worked with Paul Fasani, and he worked on dopamine at that time, and that's probably some of the early studies he did to spur his research into this field as it exists even to today. And that was in collaboration also with a Nobel laureate, Paul Greengard, over at the Rockefeller Institution, or University. He went on to go back to Italy for his faculty positions, and he's currently at the University of Campania, Luigini Vanvitelli. And his work has produced many publications, over 100 publications in very predominant journals. He's developed a program that's both basic research, looking at mechanisms of molecular actions, as well as translated this into humans. And I think we can all appreciate that that's something that we try to achieve here as well. And without further ado, he's going to talk today about the new insights of enigmatic role of D-Aspartic acid in metabolism in the adult, in the mammalian brain, from preclinical studies to clinical applications. So welcome.
Thank you very much, Daniel. I'm really thanks for your kind invitation, and I'm really delighted to be here. And you have a wonderful center of research. It's really very amazing. And most of all, I have great admiration for your mission to help people suffering for brain and spinal cord injury. So what I'm going to do today is to share with you an overview about these D-amino acids. It's actually an enigmatic D-amino acid because it's not so well characterized. Differently to the serine, the aspartic acid, it is a D-amino acid, and as for the serine in the past, it was considered just a byproduct of bacterial physiology, and therefore, in the last 10 years, we have tried to shed light on its still unclear role. So basically, as you know, all amino acids except for glycine have a chiral alpha carbon center, and therefore they exist as 2 different enantiomers, L and D. But only the L amino acid can be incorporated in the protein synthesis, and therefore, at the beginning of their discovery in living organisms, the amino acids were quite quite enigmatic molecules, not very clearly understood at that time. Indeed, in the past, they were just thought to be exclusively related with bacterial life. But in the more recent decades, and more recently, thanks to the advantages of analytical methodologies, able to dissociate, to allow it an enantioseparation, it becomes clear that also in the mammals, the amino acids exist. Among the different amino acids, basically the 2 most abundant amino acids are the serine and the aspartic acid. The serine is extremely abundant throughout the entire lifespan, although it appears to be consistently enriched at postnatal life. Differently, the aspartic acid display a very peculiar expression. So indeed, this amino acid, it's restricted the amount, doesn't. There is something wrong with the. Yeah, it works. Finally, yeah, good. Thank you very much. So basically, differently to the serine that's abundant throughout the life, D-aspartic acid appear to be restricted and to be enriched only during embryonic and fetal life. This makes me quite interested. Why that it exists a D amino acid selectively enriched in the embryo life? This observation came out from studies in '93 from Professor Hashimoto that by analyzing the fetus and the frontal cortex from fetus in the humans, reported that the aspartic acid display a highest concentration in the first trimester of embryonic life, as you can see, to then drop at a very low level at birth. And during the adulthood, this amino acid is actually maintained at very low concentration. So I would like to take your attention. So because if we look carefully here, we observe that in the first trimester, the D-aspartic acid reached almost the 50–60% of the entire pool of aspartic acid. And this is quite fascinating because why the L form of aspartic acid is involved actively in the protein synthesis. The question is why the nature has generated such D amino acid selectively at the embryo stage. This is actually one of the main questions that are at the basis of our work in the last years. Two American scientists, did a huge work in characterizing D amino acids. Solomon Snyder from Johns Hopkins Medical School and Joe Coyle from Harvard Medical School. So basically, in this case, I showed that in the lab of Solomon Snyder, Herman Volosker, at that time he was a postdoc, replicated in the rodents what was clear in the human brain. So basically, by HPLC, he showed that the aspartic acid decreased considerably during the postnatal life. And this HPLC experiment was mirrored by immunohistochemistry against the aspartic acid. And as we can see, its occurrence, it's abundant at P0, P2, P7, to then decrease and almost disappears at post-weaning stages. How to explain such time-dependent reduction in the aspartic acid occurrence? So, I would like just to remind you that while it is clearly established the existence of the catabolic enzyme, the enzyme named the aspartic acid oxidase, the aspartate oxidase, named also DDO, as the unique enzyme able to degrade at peroxisomal, in the peroxisome, actually, DDO, it's able to oxidize the aspartic acid in alpha-oxaloacetate, hydrogen peroxide, and ammonium. So while it is long established the existence of an enzyme able to degrade the aspartic acid, it's still unknown in the central nervous system the enzyme that it's able to synthesize the aspartic acid. So in a hand, we don't know the source of the aspartic acid. Conversely, we are very well aware about the pathway and the enzyme that degrade this D amino acid. So, in light with the time-dependent decrease of the aspartic acid that we have seen just a few seconds ago, we can explain that the reduction during the postnatal life of the asp is due to the fact that Van Veldoven and colleagues demonstrated that the aspartate oxidase actively degrades its substrate in an age-dependent manner. So in other words, we have that the expression and the activity of this enzyme mirror the decrease and the disappearance of the aspartic acid. So basically, it appears, the aspartic acid, it disappears. due to the existence of such enzymatic activity. Pioneeristic work in the 80s, so it means many years ago, have demonstrated that the aspartic acid is able to bind L-glutamatergic sites of NMDA receptor. So pharmacologically, the D-aspartic acid is able to bind the same glutamate sites for the NMDA receptor. Second, it has been shown that the aspartic acid can be released under a calcium-dependent manner, so as the glutamate, as a neurotransmitter. Third, the aspartic acid can be reuptake by the same AAT transporter. So it means that excitatory amino acid transporter 1 and 2 acts and work in a stereoblind fashion. Therefore, they don't recognize the difference between the two enantiomers, and they take up the aspartic acid and they remove from the synaptic cleft. So, let's start with, I would say, the present work. The first question is, what happens if we manipulate the brain level of the aspartic acid during the adult life? In other words, what is the influence of non-physiologically high level of the aspartic acid in mammalian brain function during adulthood? So this is a very important point. The idea is to manipulate differently what the nature has created. And we administer it by tap water, or simply by EP injection, the aspartic acid. And we observe the effect in a mature brain exerted by this embryo-enriched molecule. Can I revert aging processes? Can I somehow modify the glutamatergic system after such supplementation? But before to get into the detail, I want just to show you the pharmacological properties that actually we have demonstrated before to move to the in vivo studies. And accordingly to the pioneeristic work of Matus and Fag in the 90s, we demonstrate that the PAF application of the aspartic acid in the hippocampus, or in the striatum or medium spine neuro, or in the midbrain in dopaminergic neurons, is able to induce inward currents that were antagonized in a very clear dose-dependent manner by adding an NMDA antagonist. In other words, we administered PAF application of the aspartic acid on slices, hippocampus, striatum, midbrain, and we triggered inward currents that were antagonized by NMDA receptor antagonist. The question is, it's a selective NMDA endogenous agonist? The answer is not. It's not like the serine. Indeed, differently to the serine or NMDA as well, the aspartic acid is able to trigger residual currents that were not stopped, not inhibited, even at very high doses of NMDA antagonist. In all of these settings, we always seen residual non-NMDA receptor currents triggered by the aspartic acid. We will see later the origin. But it's very important to, first of all, demonstrate that the aspartic acid is able to cross the blood-brain barrier. So, to move from simple studies on brain slices to something that actually is more related to in vivo studies, we were obliged to demonstrate that the aspartic acid was able to cross the blood-brain barrier. In order to provide an answer to this question, actually, we did an in vivo microdialysis studies. In other words, we injected animal by the aspartic acid, or we treated the aspartic animal with the aspartic acid orally, and we measured directly by inserting a probe in the prefrontal cortex the extent of the concentration before and after the IP or oral administration. And as you can see very clearly, we can observe that D-aspartic acid administration trigger a very, very power increase, a very strong increase in the extracellular concentration of D-aspartic acid in the prefrontal cortex of freely moving animals. So this experiment have been done in a freely moving animal. So it's something a little bit different compared to the other studies that in the past were just restricted to homogeneous determination. Now we are characterizing the ability of this D-amino acid to cross the blood-brain barrier and to exist at the extracellular level, where it's supposed to act on an NMDA receptor. But surprisingly, and this is probably the most interesting point, is the fact that the aspartic acid supplementation or administration, besides increasing the level of the aspartic acid itself, they were able to increase the level of the main excitatory neurotransmitter in the CNS, the glutamate. So we inject the aspartic acid, and we observe an increase in the glutamate in the same brain region in the cortex. And what found by P was replicated in animal orally treated with the aspartic acid. And we can observe a consistent increase in the glutamate extracellular levels. How to explain this double effect? So, to answer to this question, we perform synaptosome preparation studies. So, in other words, we try to characterize which receptor were related to the ability of the aspartic acid to trigger the release of glutamate. And according to this study, we observe that the aspartic acid induced a strong release of glutamate that were antagonized just partially by NMDA antagonist, and was fully blocked by concomitant administration of MK, an NMDA receptor antagonist, MTAP, a metabotropic receptor antagonist, eccolo qua, MTAP, and as well by Cinequix that acts as an AMPA antagonist. So basically, we have demonstrated that injection or administration of the aspartic acid has a double effect. In one hand, it acts directly on postsynaptic NMDA receptor, but on the other, it is able to stimulate, to trigger, probably this second effect, it's even more important, the release of the most important excitatory amino acids. So, according to this basic observation, we develop a very simple strategy to increase the aspartic acid level in the brain and to investigate on its in vivo function. So, according to such modulation of glutamate system and directly activate an NMDA receptor, it was not surprising to observe that the aspartic acid supplementation as an orange juice for a few weeks was able to trigger a considerable increase in the brain activity in the regions that are very important for cognition, like dorsolateral prefrontal cortex, hippocampus, as you can see, and medial prefrontal cortex. So overall, the oral administration of the aspartic acid was translated in increase in the brain activity measured by fMRI in a brain area that are quite important for cognitive function. And very consistent with this observation, we reported that the same period of treatment, just 1 month of the aspartic acid oral administration, also increase and affect deeply the synaptic morphology, because was able to increase the total dendritic length, increased by the Scholl observation, Scholl analysis. The intersection means that the aspartic acid not only increased the length of dendrites, but also favors the interaction and the intersection between them. And last but not least, the aspartic acid directly acts on the spiny density, where the neurons make a kiss. So basically, this observation was restricted to the prefrontal cortex on the hippocampus, but we do not know what happens in the spinal cord. We do not know what happens in the cerebellum. So it was just to prove that the aspartic acid, such embryo-rich D-amino acid, has a quite robust effect in terms of fMRI and in terms of synaptic morphology. And now, to translate such structural information in something that sounds like synaptic plasticity, we observe that the aspartic acid enhances very, very, very strongly the LTP, the long-term potentiation, in a drinker's animal compare the control. And we also demonstrated that the enhancement of the LTP observed in the drinker's animal was based on the modification of actin polymerization. Indeed, administration of cytochalasin D produced a normalization of such LTP. And in line with an increase in synaptic strength in the synaptic morphology, was not surprised to find out that administration of the aspartic acid improved the cognitive function in the drinker's animal when compared to the controls. Indeed, in this drinker's animal, this animal did a very good performance, better than in the untreated animals, in what we call search quadrant, and therefore in the spatial mapping. So it is interesting that this observation, but what is its potential translational value? To answer to this question, we decided to focus our attention during the aging, because it is well established that as the brain aged, the NMD receptor overall decay, the NMD receptor transmission and plasticity is quite sensitive to the age. And therefore, we have decided to supplement animals with the aspartic acid at old stage of their life. And we have decided to use a female. Why? Because it is, again, well established that estrogen make a very important role in the expression of an NMD receptor. And after the menopause in the women's, but as well in mice and in rodents, the synaptic plasticity, the NMDA-dependent synaptic plasticity, decreased very, very, very rapidly. So we decided to take all the female, and we administered females just for 1 month with the aspartic acid. And what we found that 1 month of oral administration of the aspartic acid, while increased the aspartic level almost for twofold, even less, the effect observed in terms of LTP, in terms of synaptic plasticity, was quite robust. This is the LTP, the long-term potentiation in all the treated female when compared to the sisters untreated with the same molecule. Oh, it's very interesting. The aspartic acid has a kind of juvenile effect. This is interesting. And this appears even more clearly if we consider that animals at the age of just 2 months display an LTP that was even lesser, was even reduced compared the old female treated with the aspartic acid. In other words, we demonstrated that a short-term supplementation of the aspartic acid has a power effect in terms of restoring synaptic plasticity deterioration. And we did another experiment. We treat animals for almost 1 year. So we take 2 groups of black 6 animals. 1 group was treated just with water, was administered with water, and another one with the aspartic acid, with the same procedure that I show you so far. And interestingly, what we observe by analyzing a test called fear conditioning, that the aspartic acid drinkers showed the same level of memory compared to younger animals at 3 months of age. Conversely, as expected, during aging, we observe in untreated wild-type animal a significant decrease in the Q test retention test memory. Again, we also try to exploit this observation by analyzing the cognitive dysfunction observed under neuropathic pain. And we observe in a spare nerve injury of sciatic nerve, actually, we observe that a simple administration of the aspartic acid for 1 month from the beginning of the lesion actually was able to improve the motor coordination of treated animals. And most interestingly, the administration of the aspartic acid, when compared to amilotriptyline, that it's an antidepressant but commonly used in neuropathic pain, we show that the effect of the aspartic acid and amilotriptyline at 10 milligram per kilo showed the same effect in improving the cognitive function as tested in Y-mates to analyze the working memory. But as well, this effect was also replicated in an object recognition test. Overall, we showed that aspartic acid improved cognitive function during aging or under a brain injury. So the question is, does it is restricted to mice, this effect, or it could be translated to other species? And we start to observe in another lab, they did a very nice work in Japan, and they showed that the aspartic acid very fastly by ingestion, by ingested the aspartate, actually they showed that after 5 minutes, after 10 minutes, the aspartic acid strongly activated, again, the cortex, the hippocampus, the basal ganglia. So in other words, the aspartic acid crossed very fast the blood-brain barrier, and it is able to activate synaptic circuitry that are sensitive to NMDA receptor stimulation, those that are important for social behavior or for cognitive function. And again, what they found, and this is an experiment quite surprisingly, because they did a different study compared what I show you. I did an oral administration for 1 month, and then I've measured by Golgi staining the spiny density and so on. They did a very different experiment. They take brain slice from the hippocampus, they administer the aspartic acid on brain slice, and they observe a very powerful effect in terms of spiny density. So basically, they showed that a few minutes after supplementation on brain slice, after 15 minutes, the spiny density increase. And such increase was abolished by, again, an actin polymerization inhibitor. So overall, we have demonstrated that the aspartic acid not only bind but also activate the NMDA receptor, cross the blood-brain barrier, it's released in a calcium-dependent manner. The aspartate induces a glutamate release by stimulating presynaptic NMDA and McGlu receptor, and oral administration of the aspartato enhanced long-term synaptic plasticity and cognitive function in adulthood. And basically, what we observe, it's a beneficial effect in activating neurotransmission, in activating glutamatergic neurotransmission. But again, all these studies were executed just in a preclinical model. Is it possible to translate this observation in a human? And the answer is probably yes. And in collaboration with Dr. Bertolino in Bari, what we found is the observation about the existence of a specific SNP for DDO. This study was performed on 268 patients in the brain, or post-mortem brain, and we identified the existence of one SNP, an intronic SNP, that predicted the levels of DDO mRNA expression. So the CC genotype predicted reduced DDO mRNA expression, and probably an increase in the aspartic acid content. This study performed in post-mortem subject was then translated in healthy subjects. And by fMRI, we observed that CC genotype, that one that predicted lower level of DDO, actually showed the greater prefrontal gray matter volume. And the same genotype also enhanced working memory and brain activity as measured by fMRI. Right now, we are testing in old people the effect of the aspartic acid supplementation, since the aspartic acid is approved and commercialized for use in human as a supplement, dietary supplement. Actually, nowadays, we are trying to see what happens in the people, in the aged people with or without MCI or Alzheimer's disease. Question. It's nice, but still it's strange. Because why nature has created enzyme as the D-aspartate oxidase that during postnatal life remove the aspartate if increased brain content of this amino acid improves synaptic plasticity and cognitive ability? Why should exist such enzyme? So to answer to this question, we take advantage of mouse genetics. We have generated many years ago with my friend and colleague Francesco Enrico and my first postdoc. Actually, we have generated an animal model without DDO. So in other words, we removed the D-aspartate oxidase. We have generated an animal model that has the same content of the aspartic acid throughout the entire lifespan. So in other words, we generate adult animal that display embryo-like profile of the aspartic acid. In this way, we can answer to the question why DDO is there and what's the role of the aspartic acid, why it disappears during the postnatal life. So basically, in the first phase of our study, in a young animal, we observe very similar observation to those reported in the drinker animals. So indeed, we observe that in DDO mutants, we have an increase in the total dendritic length, again, increased spiny density in both hippocampus and prefrontal cortex. So exactly the same that we observe by oral administration of the aspartic acid. We also observe an enhancement of the LTP. So in the hippocampus, DDO mutants display an enhancement of synaptic plasticity LTP. Again, this animal, once they were at this age, at 3 months, 4 months, they have a better memory and enhancement in cognitive function and specifically in the spatial memory. But what happens if I further investigate during the aging? So surprisingly, or finally, now the picture becomes clear, we observe a biphasic profile. So at young, this animal actually showed enhanced cognitive function. But later on, they display severe cognitive dysfunction. So the yin and yang effect, persistent high levels of the aspartic acid, almost 10, 20-fold higher compare the endogenous level, trigger an initial phase in which we can observe an improvement of cognition to then turn out in a detrimental effect, producing a deficit in animal. And this observation in the behavior was also replicated in terms of electrophysiology recordings. Indeed, at young stage, the LTP was higher than was normalized at 9 months, and at 14 months of age, the LTP was almost completely abolished. So basically, this effect was also mirrored by phosphorylation of ERK, a very important protein regulating cognitive function and synaptic plasticity, and we showed that in young animal, phospho-ERK was greater compared to the control in the hippocampus, while during the elderly stages, this effect disappeared and the phosphorylation of this important protein was significantly downregulated. And we also demonstrated that the abundant D-aspartic acids, since stimulate NMDA receptor, produce adaptation at synaptic level. Therefore, we observed that over the month, the aspartic acid triggered in the mutants in knockout an internalization of NMDA receptor that actually were analyzed in the TIF fraction. The TIF fraction means the NMDA receptor that are located at the synapses. So in other words, a kind of a counteracting adaptive response of the synapse to such excess of NMDA stimulation. But since NMDA stimulation, or even better, overstimulation, it is known to trigger neurotoxicity, we observe that over the month during aging, the increase in the aspartic acid at 6 months of age starts to induce cell death in the cortex, here shown, but also in the hippocampus and also in the midbrain, in the dopaminergic neurons. Actually, we observe an increase. First, we observe an increase in neuroinflammation, as we observe in terms of IBA1 staining. We observe a severe neuroinflammation in the DDO knockout animals, coupled to progressive neurodegeneration in dopaminergic neuron. And these studies were then replicated in cell culture, demonstrated that this effect was basically related to NMDA. Indeed, the administration of MK inhibited the effect of the aspartic acid in triggering cell death. So basically, we observed that in the first phase, the aspartic acid is beneficial, and later on, it becomes detrimental. So that's why we propose for DDO a neuroprotective role that actually protect against the yin and yang effect exerted by a stimulation of NMDA receptor. So the question is, in the past, DDO was believed as an enzyme without any function, was a waste enzyme. Nowadays, we believe that it's an important neuroprotective enzyme. So, but if it's the case, should exist a mechanism regulating its expression, must exist a mechanism at molecular level that should control the postnatal expression. And we try to investigate on this topic. So I show you that the aspartic acid decrease because there is an increase in DDO enzymatic activity during the postnatal life. But actually, what is the mechanism underpinning the existence and the expression and the enzymatic activity of DDO? Therefore, we have tried to investigate the effect in terms of DDO mRNA regulation and DNA changes in terms of epigenetic modification. And basically what we found is that first, we have demonstrated that the aspartic acid has reported in rats and in humans decrease during the postnatal life. This is actually a study performed in mouse. So at embryo stage, the aspartic acid was high, at postnatal life, decrease. Conversely, the LN enantiomers doesn't change. By using the same brain samples, we investigate on the mRNA level of DDO. And interestingly, we observe that actually DDO mRNA expression occurred in a very nice time-dependent manner. That's one that we observe in terms of enzymatic activity. In other words, we are explaining that the existence of a DDO activation was actually related to the mRNA expression that follow a very clear time-dependent profile. This experiment performed by mRNA real-time PCR was also confirmed and replicated by in situ hybridization, in which we can observe a very considerable time-dependent expression. Then we moved to characterize the effect of the NMDA methylation on gene expression. And this is the DDO gene. And we observe actually the state of methylation of the 8 CpG sites. CpG sites are the hotspot for the methylation that in turn control the mRNA expression. And we analyze each of these CpG sites. And we observe that during the embryo stage, the aspartic acid, the aspartate oxidase gene is highly methylated at each of each single CpG site, therefore inhibiting the expression of DDO. In other words, DDO gene is locked by higher level of methylation during the embryonic stage to then decrease, as we can see, the reduction over the days of DDO actually lead to the transcription of DDO. And finally, to demonstrate that this was not only a phenomenological changes, reduction of methylation, increase in DDO, we actually try to manipulate the methylation in the embryo cortex. And by administering azacitidine in the embryo cortex of animal in which DDO mRNA is not expressed, because during embryo stage we have the highest level of methylation and DDO is completely unexpressed, we demonstrate that azacitidine was able to induce an abnormal, strong expression of DDO mRNA. What's the value in the human? It's very interesting because 1 year later, of our publication in 2016, it has been demonstrated by a methylone-wide association study that DDO represents the gene among the entire genome that showed the highest association with the methylation during the aging. So this study was done on 250,000 patients. So basically, we are now in front of the last question. We started saying that the aspartic acid is abundant in the embryonic life. Disappears thanks to the expression regulated by methylation of DDO. Disappears, okay, but the aspartic acid is there. It is in our brain. It is in our brain once we were at the fetus stage. What's its role? What is involvement in the development of morphology and the brain function? In order to investigate on this point, actually this is a brain staining with D-Aspartic acid, and we see that it's abundant at embryonic stage in the midbrain and that brain. Also, in the embryonic stage before birth, A20, the aspartic acid is spread everywhere to then drop at very low level. So the question is, what is the biological meaning of such abundant prenatal aspartic acid occurrence on the brain morphology and functioning, considering that NMD receptor control proliferation, differentiation, and also neuronal migration? So what's the effect of such the amino acid? To answer to this question, we generated a transgenic animal in which we overexpress DDO during the embryonic stages. So such artificial manipulation of the brain DASP content was actually able to increase physiologically the level of DDO mRNA. This is the transgenic in the hemizygote and the homozygote state, when compared to mRNA expression in wild type. This mRNA was at the basis of such amount of protein that produced an increase almost 100-fold more of DDO activity when compared to the very low level of enzymatic activity at P3, P7, and P14. But ultimately, such transgenesis lead that differently to what happens in the wild type, in DDO mutants in transgenic animal, the aspartic acid was almost 0,000 throughout the embryo and postnatal life. So I would like to have, again, last minute, your attention. Please look at the concentration. The peak concentration of the aspartic acid during the embryo stage reached roughly the 10%. However, in the human brain, it was roughly 50%, 60%. So probably there is a kind of relationship between species and the amount of the aspartic occurrence in the embryo stage. However, removing the aspartic acid from the brain of animals, we triggered alteration that somehow are reminiscent to those reported in humans. In humans, we reported that predicted DDO mRNA reduced DDO mRNA in humans was associated with greater gray matter. Here in mouse, we are observing that overexpression of DDO was associated with a decrease in the gray matter in the cortex and in the basal ganglia. Somehow, by this morphometry approach, by fMRI, we observed that there is a kind of influence of the aspartic acid. And together with such changes in fMRI, we observed that animals developed without the aspartic acid changes in the number of parvalbumin positive interneuron in the cortex that actually they have a very important role in regulation excitatory and inhibitory transmission in the cortex. Finally, from preclinical model to human, thus, again, this observation in rodents may have some functional significance in the humans. And the answer is probably yes, because we found out the patient number 0. It means we found out the existence of a young lady with the DDO duplication. We a DDO duplication that remove D-aspartic acid in their blood level, and this young lady showed retardation, mental retardation, cognitive dysfunction, and a severe coordination problem. That's why now we are addressing the reason of these differences in terms of the peak of abundance of the aspartic acid between species that could somehow explain the difference relevance of this observation. Nowadays, we are working in Parkinson's disease, and we are trying to analyze this serine and the aspartic acid metabolism in the pathology. And finally, we recently published a paper in which we extended preclinical observation in multiple sclerosis patients, and we demonstrate that oral administration of the aspartic acid to multiple sclerosis patients induce a considerable increase in the synaptic plasticity as measured by RTMS. Again, I don't want to further go in this direction, but it has been also demonstrated, and this is the last minutes, that the aspartic acid supplementation is able to prevent demyelinization induced by coprizone and to improve the motor effect related to the model of AIA model. So together this observation and the observation that indicate that the aspartic acid induce a differentiation of oligodendrocyte precursor nowadays is at the basis of a clinical trial in 20 Italian hospitals that are treating patients with multiple sclerosis with the aspartic acid because it's a safe molecule. In the man has also a beneficial effect in terms of testosterone because the aspartic acid in endocrine system is able to induce release of LH and FSH, in turn stimulate testosterone and increase testosterone in the blood, which in turn increase the strength and the muscle. Finally, my future study, or a future study that I dream to do, is to investigate, I hope with some of you, investigate the role of the aspartic acid metabolism in brain and spinal cord injury. And why not to investigate the effect of supplementation of this amino acid under such insults. My labs, my friend that actually helped this work that lasted 10 years. And so Francesco Rigo was a postdoc, was at the end of his PhD, that followed me in this adventure. And Tommaso now is taking care about the human analysis in the multiple sclerosis, in SLA, and many others. And this is the list of collaborators that I would like just mention. Subramanian Srini, that is with us, that actually is a very good friend of mine and allow me to work in the Scripps Research Institute and to work together and to grow up in the United States with you. Thank you very much for your attention. Bravo.
But you would expect, wouldn't you, that you would see everywhere? Why only in specific areas? There are some areas of cortex that showed nothing. And I would think that if it was increasing the release of glutamate, that you would see it everywhere. So why would there be a differentiation?
Okay. They asked me to reproduce your question for the recording. Basically, the question is, you expected to see a more general effect of fMRI in, I would say, in a more spread manner compared to what we observe selectively in the prefrontal cortex, in the hippocampus, and in certain region of the cortex. This is the question. So, first of all, it's very important to say that the ability of the aspartic acid to induce release of glutamate in the brain is restricted to the areas and to the brain region where the presynaptic NMDA and GLU regulation induce such release. Because I didn't show you that if I First of all, if I administer the same concentration, the L-aspartic acid, it doesn't cross the blood-brain barrier. So you have zero L-glutamate. So it means that it's a stereospecific effect. Second, if I do the same analysis, assessing the amount of glutamate in the striatum, the level of glutamate, it's exactly the same of untreated animals because in the striatum, the presynaptic regulation of the glutamatergic striatal projection is based on dopamine D2 receptor. That's why such, I would say, scattered activation for sure affect cortex and the hippocampus, also striatum. But this is actually something that is still to be better investigated. But for sure, the activation of the hippo and frontal cortical circuitry, it's crucial for us because I'm getting the aspartic acid and I can very easily perceive and have the feeling to be more focused and to be more attentive. And somehow, the aspartic acid is giving me a kind of enhancement in the cognitive function. And this is probably due to the hippocampus and the prefrontal cortex activation.
So you showed in the video of animals that later in life it becomes toxic, so neurotoxic. How do you translate that in humans?
Brava.
Whenever you have to carefully regulate the aspartate dosage, or there is an intuitive way that the brain has to regulate?
This is a fundamental question. Indeed, the question is how to explain the beneficial D-aspartic acid supplementation and how to reconcile such increase over the time once in DDO mutants you observe that the aspartic acid triggers actually cell death, neuroinflammation, and so on. So I try to stress one point during the talk. And I said that in the common animal, in the wild-type animal, you have DDO. And the supplementation of the aspartic acid for 1 month, 2 months, 3 months, 6 months, 1 year doesn't exceed 2, maximum 3 times the basal endogenous level, that it's almost undetectable. That's why the indirect effect by glutamate, most probably, it represents the driving force to affect the glutamatergic system. Because basically, the aspartic acid, it's really, really low in terms of concentration. It arrived at low nanomolar concentration, 10, 20 nanomolar concentration. It's very low. And even if you duplicate or triplicate such low level, you don't trigger the effect that you have seen in DDO mutants that the concentration were 20-fold. And 20-fold also triggered the huge release of glutamate. That's why, even though it's a matter of further investigation, the beneficial long-term effect of the aspartic acid, however, cannot be extended over, I would say, 6, 9, 10 weeks. Human studies demonstrate in the literature that they try, American researcher, actually, they try to use the aspartic acid as a modulator of endocrine function to increase testosterone and therefore the body mass and most importantly, the resistance sport. So, people that make long distance training, running long distances, were treated for 1 month, 3 months, with the concentration that are the double of what we observe and what we use in multiple sclerosis. And they conclude that actually such long-term administration doesn't affect the hypothalamo-pituitary testis axis in terms of endocrine responses. So in other words, what I'm telling you is that what it has been published in rodents in terms of the ability of the aspartic acid to increase testosterone is not translatable to the humans. Even though Merck Serono nowadays provide the aspartic acid for fertility, I am skeptic that the guys that use the aspartic acid measure only blood and muscle strength, because probably measuring their memory or their attention, probably the effect was significantly reported. I hope that was clear, the answer.
So one of the differences also with exogenous administration of D-Aspartic versus endogenous upregulation in your knockout animal is assuming that DDO only affects aspartic acid.
Right.
Is there any evidence that DDO affects other enzyme, other amino acids or anything in there? And have you looked at your knockout animals early versus late to see if other deserine, glutamate, glutamine, all those things are affected at differential time points?
Right. I replied the question. So in which extent DDO is a specific enzyme for the aspartic acid degradation, and whether during the life the absence of the aspartic acid or the increased level of the aspartic acid, the knockout, the two situation, affect somehow glutamate or the serine concentration or other amino acids. The answer is that so far DDO is known to degrade the aspartic acid and its methyl derivative NMDA. So in other words, the aspartic acid is the substrate for a methylation that convert the aspartic acid in NMDA, the aspartic acid. But the concentration of the NMDA are very, very, very low, but as well, DDO degrades both bicarboxylic acid. So this is the first question. So far, we don't know potential alternative function and role for DDO. We know that it's able to degrade the aspartic acid and NMDA, and we have proved that. The second question, in which extent overexpressing DDO and therefore eliminating the aspartic acid, or removing DDO and therefore increasing the aspartic acid, affect the serine system or glutamate? The answer is removing DDO, we observe in specific cell brain area that glutamate is increased, as I told you before. But this serine is unaffected in the knockout, and more recently we have demonstrated that overexpressing the aspartate oxidase in DDO-OV, we remove selectively the aspartic acid, and we don't have any changes in the serine and glutamate concentration. We did in several brain areas during the ontogenesis. Last but not least, you ask, what's happened during the entire lifespan in terms of the specific ability of DDO to degrade the aspartic acid? The answer is that throughout the life, removing DDO, we have a stable level of the aspartic acid. In other words, it seems that it is the unique enzyme able to degrade this enzyme. And this is actually the answer of your basic important question. Okay, the question is very similar to Daniel's question, and the answer is the question of Dr. Subramanian is in which extent the DDO activity upon the aspartate degradation is the only function recognized acting by this enzyme. So far, we can conclude that DDO is a neuroprotective enzyme able to reduce during the postnatal life such the amino acid that has a detrimental effect if it is accumulated persistently at the same magnitude as reported during the embryo stage. But very recently, we overexpressing DDO by transgenesis. So basically, we insert DDO cassette under ROSA 2026 promoter, and we overexpress DDO 30, 40, 50-fold in the peroxisome. And actually, we observe some phenotypes that cannot rule out a possible role of such overexpression of DDO in regulating different pathways that are not directly related to D-aspartic acid. But even though we can propose such assumption, we don't have proof to demonstrate that an overexpression during the embryo stages, where normally it's not expressed at all, might affect processes that probably are not directly related to the physiological activity of DDO. Thank you very much for your attention, and sorry for my bad English. I apologize.